Hi everyone, thank you for listening to my presentation. I'm Yuen from the Faculty of Pharmacy, UKM. I would like to present one of the work in my lab, which is mainly conducted by my PhD student Amnani. So this is part of her PhD work. The overall concept of the project is on drug repurposing in oral squamous cell carcinoma. And in our study, the most promising hit that we found is Geldanamycin with the structure as shown over here. And we also found that it could act synergistically together with Cisplatin. So as we all know, cancer is a population of heterogeneous cells, whereby there could be a minor population known as cancer stem cells. These cells may not be responsive to standard chemotherapy such as cisplatin, which is commonly used in the treatment of oral squamous cell carcinoma. So the survived cancer stem cells could then undergo self-renewal, which then allows it to grow and develop into a new tumor, which then causes cancer relapse in the patients. So, and we also know that there's not many new drugs available to treat oral cancer. Therefore, testing on the existing approved drugs could provide a faster route in implementing the effective candidates into the clinical trials. Therefore, in this study, we aim to rapidly identify the candidate compounds that can inhibit the cell viability of tumor spheres derived from OSCC through a high throughput drug screening process. To make the cancer model that we test more relevant, so we have grown cancer stem cell-like cells, which are also known as tumor spheres, from different patient-derived cell lines, but in the end, only the SAS cells could grow consistently into sufficient amount under the low attachment condition in also serum-free medium supplemented with additives as shown over here. So the sufficient spherical tumors grown allow further subculturing into 96 well plates, which is then could be performed on the drug screening phases as shown later on in primary, secondary, and tertiary screening, and it's being analyzed and tested using the MTS assay. Right. So before we actually conduct the drug screening process, we actually studied a little bit on what are we actually testing on. So we did some. So this is how they look like under the microscope. So this is the diameter. So it's a little bit huge in micrometer size. So we also did some transcriptomic analysis, which is then performed using the Clarum-S assay, which then revealed that there's various genes involved in the metabolism of fat and glucose, and some of the signal transduction pathways members are significantly being upregulated. So this data is then validated by performing qPCR on the highest upregulated one, which is SPT-SSB, which is the full name known as serine palmitoyltransferase small subunit B, and also on the most downregulated gene CCL2, which is also known as the CC motif chemokine ligand 2. So you can see that the data actually tallies. And we also have a look at the stem cell markers, which is the SOX2 and OCA-04. So yes, the gene expression has been increased as shown in the qPCR model, and also at the protein level as shown through the Western blot. So we therefore conclude that the model is sufficiently cancer-ish like. Right, so we then move on with the primary drug screening. We did it on actually 1,463 drugs which are FDA approved or those who are already in the clinical trials, whereby in this phase, it's only one well being tested for each drug at 10 micromolar for 72 hours using MTS assay. After correcting their interplate variability, B-score normalization is used to filter out the hits. So the hits are they actually defined by B values lower than 3 MAD, which is whereby MAD actually refers to the median absolute deviation. So from the 174 candidates, we further analyzed using the stitch method and to recategorize them based on the mechanism of action. And there are currently available evidence which have been published in cancer studies. So we then further select 8 drugs for the secondary screening. So here we just show a part of our data that we do found that geldanamycin has the lowest IC50, which is at around 28 nanomolar, and we further test them at different concentrations with cisplatin, and we've got a combination index analysis of 0.66, which suggests that it's a synergistic effect together with cisplatin. So in relation to the mechanism of action, geldanamycin actually binds to and inhibits the cytosolic chaperone protein functions of the heat shock protein 90, so which is HSP90. So it's originally discovered as an antibiotic. So it can actually promote proteasomal degradation of oncogenic signaling proteins, which may be overexpressed or overactive in the tumor cells. However, geldanamycin is only in the clinical trial phase, so there's further evaluation can be done with geldanamycin and also with the newer members of its group, such as Cisplatin. And we can also test the interaction, the mode of interaction with cisplatin, and also further test its in vivo efficacy as well. So that's all for my presentation, and I will also use this opportunity to thank the funding body, which is mainly by my university, UKM, and also thanks a lot to my collaborators within UKM and also outside UKM in IMU as well. Thank you for your attention.